Welcome to the NanoCap podcast. I'm your host, Will Turnlin, and I'm excited to have you join me on this journey through the world of NanoCap companies. For the past decade, I've talked to thousands of business owners across various industries. It's been an enlightening experience revealing the vast tapestries of entrepreneurship and innovation that thrives beneath the surface of mainstream media coverage. Each week, I will interview different leaders of NanoCap publicly traded companies to learn about their history, what they do now, and their future. As a reminder, none of this information is financial advice, and it is all for entertainment purposes only. Let's start this week's episode. Hi, everyone. This week we have Chris Burns, the CEO of Amplia Therapeutics, stock ticker INNMF. Amplia's lead drug candidate is a highly selective and potent efficacy inhibitor that has promising results in studies for treatment of pancreatic cancer. Chris, thanks for coming on the show.
Happy to be here, Will. Lovely to meet you.
So, Chris, as our viewers might have, or listeners have already kind of maybe figured out, is that you originally are from Australia. But as I was kind of doing a little bit of digging, I would love to hear a little bit about how you ended up at Happy Valley in Penn State and kind of what it was like culture shock wise. People come to America, they come to LA, they come to New York. It's not the real America. There's basically no more real America than Penn State in Happy Valley in Pennsylvania. So tell me about the culture shock. Tell me about how you ended up there. I'd love to hear a little bit about that in your research journey.
Sure, absolutely. I've never been asked that question before, so that's great. Happy to answer it. Yes, I'm a Penn Stater. Still follow the football as much as I can. My background is in chemistry. I did a PhD in chemistry in Australia and wanted to do postdoctoral studies, and looked at a number of options for that and really liked the program that was being run at Penn State. I worked with a really talented chemist by the name of Ken Feldman. And I wrote him a letter. I'm so old, this is pre-email. So I wrote him a letter, and about 4 weeks later, I got a reply. That's how things used to work. For the older people in the audience will appreciate that. And he offered me a position, which was fabulous. So I flew over there in October, and I've never been as cold in my life as I was when I landed at Penn State. And I think I was unlucky. I came at a really cold snap, and that was probably the coldest it was for the next 2 years that I stayed there. Look, it was a reasonable culture shock, I guess. I couldn't believe how many people are into college football, but got very much into it myself and thoroughly enjoyed it. I still have close friends from my time there, which was, some 30 years ago now. So, had a great time, met some really fabulous people, and learned a lot of really cool science.
Yeah, no, I can just imagine you getting there as the leaves are turning, as it's middle of football season. They just won a national championship like 3 or 4 years before you got there. So it's still the middle of the Joe Paterno era. And you coming in with, like you said, no internet access. So it's not like you have like an encyclopedia you can read in Australia to get caught up on Penn State culture ahead of time. You kind of just get dropped into it, and all of a sudden you learn all about the Nittany Lions.
Exactly. Yes, learn a lot about the Nittany Lions, and thankfully, the way labs work, research labs work, is that you become, a close knit group of friends and family. Friends that become like your family. So all my colleagues there at the time were very pleased to introduce me to life at State College, Pennsylvania, and how to cheer at the football and go to a tailgate party and all that kind of stuff. So yeah, had a lot of great memories from that time.
A quick dumb follow-up from my point of view. Is there any sort of difference when you're doing research over there with like the imperial system versus the metric system, or is all science just done in the metric system already?
Yeah, I think all science is done in the metric system, thankfully, because I've got no idea about an ounce and a pound or gallons. That's all a mystery to me. Yeah, liters, kilograms, that kind of thing. So thankfully, we do all that stuff in metric around the world.
So you escape the Midwest with your research, and then you're able to get back to Australia, and then you're still kind of, for about almost a decade or so, still in the research realm. You could have spent a whole lifetime researching and hanging out in academia and different labs and such. but you wanted to jump into the kind of business side of things. Was that something that you always kind of had a burning passion for, to be like, okay, I want to create and be in the business side of things? Were you kind of pushed into it over time, where you go, hey, there's a great research opportunity and there's great business opportunities? Or kind of what made you want to make the flip?
No, that's a good question. There is a break in my career between leaving Penn State and getting back to Australia, and that was 5 years in the UK. I moved to the UK and actually worked in industry there, worked for Pfizer. So I had that taste of what drug discovery and development was like in a big organization, and really loved it. Worked with some absolutely fabulous people over at Pfizer. Moved back to Australia for various personal reasons, and there's not a large, drug development industry in Australia. So ended up back in academia for another sort of 5 years, I guess, but always wanting to get back into drug discovery and development. And I was fortunate that an opportunity presented itself. I joined a company that at that time was just starting up, just building a chemistry group. The company was called Cytopia. We were really successful in the whole field of kinase inhibitor drug discovery. That was, in the early days of the development of kinase inhibitors in cancer and beyond. And from that, we discovered a drug that ultimately got approved as now a marketed drug. So I was part of that process, part of being in a small biotech and growing that. And I guess I really love that challenge. So I like the sort of cut and thrust of being in a small company where you're looking at pushing a program forward as quickly as possible and being able to respond to data very quickly. That's a little bit different than being in a big organization sometimes where there's a protracted and risk averse decision making. Biotechs, you tend to be a lot more agile.
So, yeah, tell me a bit more about that. What about doing research in Australia? You said some personal things, but is there some benefits to doing some sort of biotech research in Australia that they don't have in America, and vice versa?
Look, if I'm wearing my finance hat, I would say that one of the key benefits is that it's cheaper to do research in Australia. Staff costs are not at the same level that they are in the US. I think Australia and the US are very similar in terms of the quality of the science. You have really excellent scientists in the US, and obviously that's why I went over there to study myself. But we have really strong medical research here in Australia. Grew up in Melbourne. I returned to Melbourne. Melbourne probably is a bit like Boston. It's slightly warmer in winter, but we have a lot of really world-class medical research institutes that are all very close. There's a lot of cross-pollination there, and work closely with universities and research institutes in the US and Europe as well. But so it was coming into an environment that it's not dissimilar to what many scientists in the US would know and love about, say, Boston or the Bay Area or San Diego, all those other sort of hotbeds of both excellent science and then biotech that builds around that.
100%. So you worked at a few different research labs, and then you kind of get into the private side of things and start working for some corporations and such. When did you come across Amplia? What was the first time you heard about it? And with all of your experience pattern matching and seeing all these different people trying to solve different types of problems, why is this the one that kind of stood out to you as like, okay, this is where I want to spend my time?
Yeah, it really comes back to the drug itself. The main drug we're developing is a FAK inhibitor called Narmaphonib, and this drug was discovered at a industry academic partnership. It was sort of funded partly by the government, partly by industry, and that group, which was called the Cooperative Research Centre for Cancer Therapeutics, so a bit of a mouthful, they had this really core group of experts in drug discovery, and they were working with institutes that had a lot of excellent biology and allied skills. And they discovered this class of FAK inhibitors, and I was living and working in Melbourne. and I became aware of the drugs, and I thought these were really good quality molecules. They had some really interesting activity. And in about the mid 2000, so around 2015, 2016, the mid 2010s, research started coming out in the literature showing that if you inhibit FAK, you can actually improve the outcome, at least in animal models, of in certain models of cancer, and particularly by modulating the fibrosis around tumors, which is like the scar tissue that sits around a lot of solid tumors. And also you can modulate the immune microenvironment to make other drugs work better. So that was really exciting to me. I could see that there was a potential, and it was really sort of a lot of companies were looking at ways that you could improve the activity of immuno-oncology agents like Keytruda, for instance, that we know is, that's been a massive blockbuster for Merck. And we thought, well, certain tumors don't respond to these immuno-oncology agents for various reasons, and a FAK inhibitor might be able to make those work better. And we started seeing some publications out of the UK and out of the US that were showing that. So I got together with a couple of other scientists, actually, and we founded Amplia. So it was really on the back of the science that was being published, and knowing about the assets that were sitting here at the Cancer Therapeutics Cooperative Research Centre. We thought, well, let's put those two together. Let's form a company and try and progress, what we believe is the best asset that's sitting in the Cancer Therapeutics Cooperative Research Centre. And so Amplia was born, and then we started progressing the development of Narmaphonib.
And so kind of explain it to me like I'm 5. Break it down. So when there is someone who has a tumor and they're doing some sort of like therapeutic or chemotherapy, would they automatically want to use your drug on top of it, since it would just make the chemo that much more effective? Or is it something where they go, okay, we've tried chemotherapy a couple times, it's not seeming effective, we need to bring in the big guns, we need to bring in something to kind of bust down this wall so the chemo can get through to it? Can you kind of explain a little bit more, just layman terms?
No, that's a great question. Look, we want to position Narmaphonib as a drug that can be used in combination with a whole range of different therapies, be it immuno-oncology, be it chemotherapy, be it targeted agents. But because the way an FAK inhibitor works is it does target this scar tissue that sits around a lot of solid tumors. So pancreatic cancer, head and neck cancer, lung cancer, breast cancer, they often form a scar tissue that really stops the immune system from being able to get in and attack a tumor. It stops drugs, and particularly biologicals, from getting in and penetrating into that tumor and killing the tumor cells. So there's that benefit that we see that a FAK inhibitor could have in a range of solid tumors. You're just allowing better access into the tumor tissue for the immune system and for drugs. On top of that, though, that an FAK inhibitor also blocks processes that are happening within the tumor cell itself. So in the tumor cell, you'll often get FAK will be upregulated. It'll be at higher levels than normal tissue, and it'll be driving processes that lead to cell survival, cell proliferation, and migration, in particular in response to cell stress. So if there's cells already under stress, you can get an upregulation of FAK, and stress could come from radiotherapy, it could come from chemotherapy. So FAK is often upregulated in response to other drugs, so it becomes a resistance pathway as well. So we see, and what I like to say in these kind of discussions is that a FAK inhibitor acts as a bit of a 1-2 punch on the tumor. The first punch is really affecting that microenvironment, and the second punch is a punch on the tumor to actually stop some of these processes that actually allow the tumor cells to survive and grow, even in the presence of chemotherapy. So to go back to your original question, we think, and the work we're doing now is really to combine Narmaphonib with the therapy from the get-go, not sort of wait until third, fourth line treatment, but to actually have it there from the outset, always making whatever type of therapy, be it chemotherapy, targeted agent, biological, making those things work better.
Amazing. And so I saw an interview from you about a year or so ago, and so I would love an update on where are you with the different trials? And are you trying to get it approved in Australia, UK, the whole Commonwealth? Are you trying to get it approved in the US first? Kind of what's your strategy there? And does getting approved one place make it easier to get approved other places?
Yeah, so look, our focus is very much building a data set for the molecule, initially with the goal of driving for approval in the US. So we have a close interaction with the US FDA. We have orphan drug designation in pancreatic cancer and indeed fast track designation in pancreatic cancer. So both from the FDA. So our focus is very much in the US. We've had two clinical studies that we've run to date. The first has been a study combining our drug with standard of care chemotherapy in pancreatic cancer, and we're running that study here in Australia and in Korea. So we've got some really promising data, which I'll come back to in a minute. And then secondly, we've started a trial now in Australia and the US under an IND. We've opened up 2 sites now in the US, I'm happy to say. And that's combining, again, our FAK inhibitor Narmaphonib with a different chemotherapy that's used in pancreatic cancer, more commonly used in the US, actually. So that trial's just starting to recruit. The goal is to build out a data set to get comfort with the way our drug plays with both different types of chemotherapy, such that we can actually map out what a registration-enabling study looks like. And that's, in fact, exactly what we're doing this year. We've got enough positive data from our ACCENT study, from the first study we're doing, to really have a good idea what a registrational study will look like. So we're planning that. We intend to engage with the FDA in the first half of this year about the fine details of what that trial looks like, with the goal of starting that trial by the end of this year. So that's, there's a lot going on in the background to make that happen. And it's really building on the promising data we've seen to date of our drug in combination with 2 chemotherapies called gemcitabine and nab-paclitaxel, sometimes referred to as Abraxane. So these 2 chemotherapies, often used in pancreatic cancer, they'll give a pancreatic cancer patient about a 5 to 6 month improvement in survival, so progression-free survival of about 5 to 6 months. We've shown that our drug can improve that by 2 months, that progression-free survival, when compared to the historical data. We've shown that we get a deeper response than what you'd expect normally for that chemotherapy, and that response is durable. And importantly, our drug, Narmaphonib, is not really adding to any burden to the patients. So these patients are sick because of the disease, they're sick because of the chemotherapy. Our drug actually has very, very modest tolerability issues. So a little bit of fatigue, maybe a little bit of nausea, all manageable in the broader context of the disease. If we look at the severe side effects of chemotherapy, when you add our drug on top of it, it's not changing that at all. So we're not adding any additional burden to these patients. So that's sort of the 3 things. The drug has improved efficacy, improved durability of response, and improved tolerability. And I might add that our drug is given as a pill. So there's this convenience aspect as well. So you can take it as a pill. It doesn't interact with other medicines. So we're ticking a lot of boxes there. So on the back of that exciting data, we're planning now for the registrational study to start, hopefully at the end of this year.
No, that was one of the things I wanted to highlight, is that it's just taken as a pill too, which is very, very convenient. So Chris, very exciting stuff, but gotta ask you kind of, what gets you out of bed in the morning? What gets you excited about this? What makes this what you want to work on? What about this inspires you?
Yeah, no, it's a great question, and it's an important question, I think. It's hard doing biotech. Anyone will tell you the drug development is complicated and challenging. But pancreatic cancer is the 3rd biggest cancer killer in the world. It'll probably be by 2030 the 2nd biggest cancer killer. Patients currently, if they're diagnosed with metastatic disease, so advanced disease, and this is the patient group we're looking at, their likelihood of being alive in 5 years is somewhere around 3%. Pancreatic cancer patients need better treatments. That's really, really obvious. We think the science is now developing to a point where we know how to tackle aspects of the disease, and particularly, with our drug, it's around this fibrosis, this scar tissue that sits around the cancer. And we think in combination with chemotherapy and in combination with other therapies that we can actually improve those patient outcomes significantly. So it's a really exciting time to be working in pancreatic cancer. There's a lot of exciting development. The whole field of KRAS inhibitors that you've probably heard about is an area of intense, competition. That's a real opportunity for us because we know that our drugs, based on preclinical work and a solid understanding of the science, that our drug will enhance the activity of those KRAS inhibitors and make them work better. And so, I can see that rather than patients looking at a 3% chance of survival at 5 years' time, that we could look in the future and probably in the next decade that could be, a much higher percentage. We're not talking about cures yet, but we're definitely talking turning the corner here, that pancreatic cancer is not such a grim prognosis as it currently is. And that's what excites me, that there's a lot to do here. There's a lot that can be done for pancreatic cancer patients, and I think our drug actually is going to be an integral part to that. And then if we think beyond that, take a step back, this sort of idea of these heavy scar tissue that sits around the solid tumor is not just in pancreatic cancer. We can think about ovarian cancer, we can think about lung cancer and certain types of breast cancer and so on and so forth. So there's opportunities, beyond that as well. So it's really exciting science that we've got. The clinical data's been very promising to date, and we hope to build on that going forward.
Well, Chris, thank you so much for coming on the show today. If my listeners want to learn a little bit more about Amplia Therapeutics, where should I send them?
So the website's Amplia, so it's like amplify, because we amplify the effect of other drugs. So it's AmpliaTX for therapeutics.com. So AmpliaTX.com, and there's contact details. You can sign up to our shareholder announcements, and you can see a monthly video from myself and my COO. We give a monthly update on progress, so that's something to look forward to too for your listeners.
Well, thank you so much for coming on the show, Chris, and everything you do to make the world a better place. And for those of you still listening, please remember to subscribe and review. Thanks.
Thank you.
This podcast is for informational purposes only and does not provide financial, legal, or any other advice. The information is not investment advice or any offer to buy or sell any securities or to make any investment. The views expressed by the guests are their own, and any reference to third-party products, services, or information does not constitute an endorsement therefor by NanoCap Podcast or its affiliates. The NanoCap Podcast Express disclaims all liability for any individual's use of the information presented in this podcast.